As always, we start with our bell ringer, and today we have a sex-linked recessive trait. Hemophilia is a sex-linked recessive trait. A mother with normal blood and a father with hemophilia has a daughter with hemophilia. Which statement is correct? Now with this one, we're going to have to work our way backwards. First, we know that the father has hemophilia, so we're going to put a little h up here to represent the father. And we know the mother is normal, but we don't know what this one's going to be, so we're just going to put one capital H here. And then it says has a daughter with hemophilia. So we know one of the daughters is going to have a little h, two little h's. So when she has the two little h's, we know this person or this mother right here had to have the smaller h also, so she has to be a carrier. We'll go on through and make our cross. So from this Punnett square, when we look at it, we know that all of the daughters have hemophilia. Well, this isn't true because this one's a carrier and this one has it. All of the sons will have hemophilia. Only one has it. The mother is a carrier of hemophilia. We know that's true because she has the lowercase h here. So that means she's a carrier. And then it says only one daughter out of their children will have hemophilia. That's true also. So we have two statements that are true. That is true, but this one is a carrier. So we know this is true. So for this one, we could have had C and D as the correct answer. Now, we're moving on from sex-linked traits, and we'll be going on to recombinant DNA. All right, remember yesterday we talked about restriction enzymes and how they cut at specific sites on DNA. And this is one that we did yesterday, H3, where it forms blunt ends and it cuts the DNA strand. Today we're going to be talking about recombinant DNA technology. And with that, we start off with a plasmid, which is circular. This is a circular form of DNA, and we remove the bacterium. DNA with the gene of interest is moved to a donor. So we end up cutting it with the restriction enzymes, as we spoke of yesterday. We cut the DNA and it leaves out sticky ends. Remember we said sticky ends was where they're going to cut and leave little formations like this to be able to be inserted back into the complementary strand. Recombinant DNA contains DNA from two different sources, and one source is the bacterium and the other source is a human. And the reason why we use the bacterium is because, like in the case of insulin, we need mass production of insulin. And it's not just like when we do blood and ask people to donate and we do blood drives. You can't do an insulin drive. So for those with diabetes, we have to use something else, and that's why we use the bacterium, because it reproduces more. When we transform the bacterium, we take the recombinant DNA and we insert it back into the host. So this one is E. coli. And the bacteria becomes transformed, means it changes and becomes something new. And it produces a transgenic bacteria. And the organism is formed with the DNA. So this is what takes place when we cut it and make the recombinant DNA, which means we combine the DNA to make a new plasmid. We insert it back into the host, which results in a transformed cell. In order to have the cloning transgenic bacteria, we must first, as we said before with the process, we use the restriction enzyme and we cut it to form the plasmid. We insert the human DNA in, which is the complementary DNA, and we put it in with the linkers. The bacteria cell clone the gene of interest, and when it clones the gene of interest from the bacterial plasmid, we have insulin. And it's used to make mass amounts of human insulin or human growth hormone. And that's the purpose of us using bacteria, is because it can reproduce over and over and over so quickly, and transfer and cloning of the insulin DNA takes place there. And the application is, of course, like we stated earlier, it produces insulin. So when all the process takes place, we have the bacteria, we isolate that, we have the cloning cell and the plasmids into the bacteria. And then the cells multiply and grow, and we have multiple copies of them, and then we can place them back into humans, and they can use that for insulin production. Now, the production of human insulin, it regulates the blood sugar level. The gene for human insulin is inserted into the yeast or the bacteria, from which large quantities of human insulin are manufactured, and this is what takes place as we do that. We also have to use this for when we make other genes that we need for other production of other